Then good afternoon to everyone and good morning for me. It's 8 o'clock in the morning here in Germany, and it's a great honor to participate to your seminar series. And today I would like to talk about some work I did, some mathematical work I did to try to understand brain computations underlying perceptual decision making, and in particular two fundamental operations that need to be done in order to take actions of what you perceive, that is to essentially encode information about the sensory world and then read it out, accumulate evidence to take decisions. And I'm a computational neuroscientist, so of the work that I will present, I am my lab and all the postdocs and that contributed are listed here. That did most of the work, of course, produced the mathematical algorithms and analysis, but the data, brain data that we use, I mean, to motivate the algorithm and also then to find results about the brain are from collaborating experimental laboratories. Most of them are from the lab of Christopher Harvey at Harvard Medical School, and some of them are from Dima Rimberg at NYU, Karunesh Ganguly at UCSF, and Tobias Donner here in Hamburg. And the problem essentially we try to study the brain function is a perceptual decision. So this is a fundamental operation. It's essentially what many machine learning algorithms do. Essentially, you take an input, the brain takes an input, and decides what the most appropriate action given the input. And in many cases, this input may be a sensory input. For example, you see something and you have to decide what to do with it. For example, do I eat it or not? And that would be essentially a behavioral output or a decision that you take based on the input. And what essentially allows you to do this is brain activity, shown here as a simple movie of the fluorescence of cells that correspond to brain activity recorded with 2 calcium imaging. And essentially, one of an important question in brain sciences is how the brain does this operation. So taking information from the senses and then elaborating it to take an appropriate action, an appropriate decision. And our work as a computational neuroscientist is to try to develop analytical methods for dissociating the two operations that at least two operations need to be done to do this. One is sensory encoding, mapping sensory stimuli into neural activity. What the brain knows about the world is not this, but it's the pattern brain activity. And then taking the information embedded, hidden in this pattern of activity and use it to generate an appropriate output. And so our analytical methods that we try to present in this talk try to dissociate the two operations and then to understand how they are combined to determine in what the fate, the destiny of the information encoded in the considered set of neurons to determine behavior. And the take-home message in this talk is that these two operations are often dissociated, and maybe what the brain tries to do in designing neural codes that can perform this action is a trade-off between the different needs for encoding and readout of information. Um, so as we start exploring this problem, I think the one that has been studied the most is the problem of encoding of sensory information. And many people, essentially, also especially in sensation, of course, have studied this. And sensory encoding is the map between the external world, for example, images, sounds, odors, whatever you like, and neural activity. So the code, the alphabet that is used to encode in the activity of the considered brain area about the external world. This is called sensory encoding. So it's the forward map between the external world and neural activity. And then also there many people have considered also what to study this for practical purposes. For example, also you can think of brain-machine interface and so on. The inverse operation, the decoding. And this decoding is conceived as the ideal reverse map. So essentially how your machine learning algorithm, for example, a Bayesian decoder or a linear decoder, whatever, would decode this. And so this can be done, for example, with information theory. Information theory would allow you to study this encoding, which is essentially how much information or how the information uh, about this is present in neural activity. And Shannon information is a tool that we will use, mathematical tool to study this, which is a function of the entire stimulus and probabilities, and captures all ways in which information can be expressed. And so it's excellent for bounding performance of any decoder, of any algorithm, and so on. And then another very popular approach of this, which is this decoding idea reverse map, is to try to use your best machine learning algorithm, your favorite machine learning algorithm, can be a linear decoder, a GLM, or a Bayesian decoder, and so on, to extract the most likely stimulus. So, for example, an apple or a pear, given the observation on neural activity. And if you use this kind of reasoning, this sensory encoding, I think it's very useful because you can answer many important questions. And one that, for example, people have studied for many years, including myself, is what is the alphabet, the code used by this population of neurons, for example, in sensory areas, to represent the external world. So what are the features of neural population activity that are important for information encoding? And there are many questions that you can study. Then we study, for example, what is the role of the timing, the time scales of neural activity for encoding information. We have done in the past a lot of work, for example, showing that short time scales, so millisecond precision, spike timing encode information for sensory perception, but then long time scales are needed for accumulating evidence over time. Other question that the one also try to use to illustrate today our algorithm is what is the role of the functional correlation between the neurons in encoding information. Whenever you measure two neurons, three neurons, four neurons together, if you try to compute whether the activity of the two neurons is independent or correlated, you find that almost always the activity is correlated. So there is functional correlation between the activity of the neuron, but what do those correlations do? Do they help information encoding? Do they instead hinder it, and so on? So those are the type of questions that can be addressed with this type of approach of sensory encoding. And but there is a danger, I think, in this, and just considering information encoding. So if you consider a given neural population and recording in a brain area, I don't know, visual cortex, and then you determine, okay, here this population encodes information, the spike timing encode information, correlation do this and that. But then you consider only the information that is there. You don't know whether this information that is there is used by the brain to produce a function. It can be that it is used, it can be that it is used, but only to some extent because of the limitations of the brain, or maybe this is completely epiphenomenal. Maybe, this is something that is not used, this information by the brain to take the decisions and so on. And our work was to go beyond this approach of sensory encoding to measuring the fate of the information encoding in neural populations. And I developed three approaches that I try to synthesize in this talk that can be adapted to different experimental setups. And one approach is so usually in many experiments, people record information either with microscope to photon imaging or with a set of electrodes only in one area, say a cortical area or a brainstem area, and so on, a thalamic area, or maybe just a small number. And as I expressed in the previous slides, they compute how much information is encoded in this local population that you record. But then, if you have this recording from one area and so on, we try to develop methods that compute not only how much information you have encoded locally, but how much of this information is actually used to inform the behavioral choices. And I will illustrate how we do this. So this can be adapted to try to understand how the brain uses information encoded, just having a recording you have just from one area. And then also, if you have recording from multiple areas, you might want also to know if you have an idea of the cortical or the processing hierarchy from essentially lower level to higher levels. Then you can determine how population in an area that project to the same that to target encode information. And we did some work on this, and experimentally, this requires being able to identify or label the neurons within a local neighborhood that transmit to the same downstream target. And the third thing that we did, if we have recordings from multiple areas, I don't know, area 2, for example, visual areas, parietal areas, and so on, we can determine how much of the information encoded locally in one area, for example, the visual cortex, is transmitted downstream to another area. And so I think by the combination of this method, I think we can determine a number of things about how information, how about the fate of information recorded in a given area, what is epiphenomenal and what is not.
So can
And
I
please,
question?
yes.
So I don't understand what is the meaning of the fate. So yeah, these three.
Fate is a destiny. So fate means what happens next, you know. So my fate is it's means that you know what will happen to my life, and
Uh-huh.
fate means what happens next. So you have information encoded, but is it used at the end
Uh-huh.
of the process when you take a decision, or is it not used? So that's what it means.
It's kind of the predictions.
No, it's not a prediction, it's an evaluation. So we are
Oh,
able to determine if a bit of information encoded in a given area in the brain is
uh-huh.
used to produce the behavior or is not used, not how
Uh,
much is used. So you could have that you see this population and you say that it's encoded one bit of information,
mhm.
but then we will conclude this bit of information is used or is not used or it's used only partially, is reflected only partially in the decision taken by the entire organism. The behavioral decision is expressed by the entire brain, the entire organism.
And
So
in
I will try to, yeah, yes.
yes, 3 point is very important for understanding the fate of the so information encoding.
Yeah.
Three, no, three, all of them I think are important in my
Ah,
view.
yeah, three is a computer how much. Yeah, you have so showing the 3 point.
Yes, yeah.
Yeah,
So in
Stefan,
my
sorry
view,
to
please,
join late. Yeah, but
I
I
can,
have a question about this fate. So the behavior in the same trial or
yes,
next trial or
yes,
behavior in the next day, what kind of
yeah.
timing do you consider?
So here in the talk today, I consider behavior in the same trial.
Mm.
So short time scale consequence.
Yeah.
So information, say perceptual decision making, I show you, I don't know, a stimulus, a visual stimulus,
Mhm.
and then I ask you to turn in a certain direction depending on the visual stimulus to collect the reward. So this is the short term fate. It's used within the same trial.
Mm.
I
Okay.
don't have time to show today, but in other work with Mrigan Kasur at MIT, using reinforcement learning tasks, we're using this kind of stuff to determine how astrocytes, for example, determine behavior in the next trials after a
Mhm.
surprising outcome.
Uh-huh.
But
Okay.
what we do today is, sorry if I was not clear, but I hope it will become clear when I give the talk, is essentially the use of this information within the same trial.
Okay. Thank you for clarification. Clarification. Yeah.
So, okay. But I hope this is an overview. I hope that this will become a little bit clearer as I give the talk. And if not, it means that the talk was not very good. But I hope that some of this will be clarified. So I try to go ahead. And so to determine this essentially whether this activity of the neurons is used or not for producing a choice in perceptual decision tasks, so choice within the same trial, as Kenji said. What people did for a long time is something that is called choice probability. And essentially, what experimenters do, as the animal does some discrimination, for example, you have to determine whether the dots are going left or right. Every now and then, you insert some catch trial with zero information. So, for example, the dots don't have any preferred direction. And then the animal, if it is an animal or a person and so on, will essentially, if we forced to take a decision, will take a decision, say this is left or this is right. And from these zero information trials, they take a map. Essentially, they compute some decoder and so on, some measure that tells how much this neural activity can predict the choice. The reason why people insert these zero information trials is that if the animal or the person, the subject, can do the task above chance, there will be a correlation between the stimulus and the choice. and you want to try to separate out those signals. And if you remove any information here, it's the simple way to do it. But in my view, at least, a limitation of this, that here we are not really understanding how information is read out. I think we're understanding how zero information is read out. And so I think there is some limitation. I think it would be better in many ways also to be able to do the same, but really reading actual bits of information about the sensory environment. And so that's what we try to do. And essentially, what we try to do, our logic for the first approach, that is taking a local area, so some population, and try to understand how information is encoded here about the sensory stimuli, and then how much this information about the stimuli is able to inform the accuracy of the choices made by the subject. And to do that, essentially, what we do will be to construct, as we do in studies of sensory coding, a map between the sensory stimulus and neural activity. So we compute the information about the sensory stimulus and encode the neural activity. And then we compute also what we call readout, which is different from this choice probability. Is not a map between neural activity and choice. It's a map between the stimulus information encoded in neural activity and the choice. You can think of you have a posterior probability of neural activity, posterior probability of the stimulus at any time, and how much this posterior fluctuates on a trial-to-trial basis with the fluctuation of the choice. And so essentially we isolate in a way the components of neural activity that carry information, and then we see how those components, not the entire population activity, fluctuate with behavior. And mathematically we did in many ways, but the backbone of this is, of course, essentially is a trivariate problem where you have to build a probabilistic model of stimulus, choice, and population neural activity. So I mean, it's multivariate if this is many neurons, but conceptually it's trivariate, that you have three classes of variables. And we did it with various tricks, either sampling the probability directly with small populations, or using a probability model to fit to the data from simple generalized linear model to more complex kernel methods and whatever. But the backbone, I think, is that essentially we have a trivariate probability model of stimuli, choice, and neural activity in the same trial. And we did this stuff in two ways, which I think are similar, give similar results onto the data, but conceptually are slightly different. One is with information theory. And the way we formulate this with information theory is that we consider this information that we have in neural activity, and we ask how much of this information in neural activity that is encoded in neural activity about the stimuli is used to inform the choices on the same trial. And the way we define this is a bit complicated, but I think it's nice because it has a lot of properties, the traditional bounds of information theory, which are very useful. And essentially, we use those partial information decomposition, which are techniques to, for example, detect what is redundant between many sources of information, what is encoded redundantly, what is encoded synergistic, and so on. And essentially, we identify the part of information that is shared between the information in neural activity about stimulus and information in neural activity about choice. So it's information in neural activity that is redundant about stimulus and choice. And the nice thing about it, we had a NIPS talk a few years ago, is that it satisfies all the bounds of user information, which are very useful, as you know, from the people, the people from you that come from an engineering background. And then there are
So
simpler
I'm
versions.
sorry,
Sorry,
can I ask a question? So let me
please,
check the basic things. So
yeah.
S is a sensory and R is a neural activity.
Yes, and the C is the behavior, the choice, the
Uh,
behavioral readout. For example, in many tasks, if you have, say, a binary stimulus, would be apple or pear, or left or right, a sound from the left or from the right. And
mhm.
then the readout is what essentially the various possibilities that the animal has to reply to this in order to show that, for example, it recognizes the stimulus correctly. For example, in the task that I show you later, stimulus would be a sound coming from the left or from the right, and the behavior is I turn left or turn right because I need to turn toward the direction of the stimulus in order to be able to collect my reward.
And I'm
And
gonna check
so,
sorry, so I'm gonna check the I is also I is a mutual information.
yes, I is a mutual information.
And
And II, what I call
yeah.
intersection information, which is a
Okay.
quantification of how much of the stimulus information encoded neural activity is shared with the choice or is used by the choice.
And the intersection information is defined by you.
By me, yes. By this
Ah, okay.
paper here, this is the new ripstock of a few years back.
Yeah, okay.
This is one way we address it. Yeah. So essentially, I mean, Shannon information was done by Shannon, and he did it very well, but is essentially totally defined only as a bivariate problem, X and Y, stimulus and response. This is a little bit more complex because it's a trivariate problem. And our contribution to this was we say, okay, out of this information, how much is read out downstream by this other variable, which is the choice. So we don't know all the other pieces that are in between. So between this activity and this, there are many neural stations and so on that we don't record. What
Mhm.
we try to do to understand the behavioral relevance of this information is to try to relate these to the total output of the system. So essentially, it's a way to try to estimate the total effect of this population, the information carried population onto this without not necessarily knowing all the pieces in the middle.
Mm. Okay, thank you very
Okay.
much.
Okay. And so one way is to do it with information theory, which is a bit complicated, but has many satisfying, at least for us theoretician, things. And another, which is a little bit more simple, that neurophysiologists like better because it's more interpretable, is to use decoders. So essentially what we do, to cut a long story short, we construct decoders in this space, multivariate space of neural activity. So for example, this is a bivariate space where you have two neurons, but you can think of this as a hyperspace with many dimensions, where every dimension is a neuron. And R1 will be the activity of neuron 1, R2 the activity of neuron 2. What you can do, you can construct decoder in this space. Here, for simplicity, we construct a sensory decoder, so a linear decoder that partitions the space into prediction. But then you can do the same also. You can train the decoder to decode decisions, and you would have another decision boundary. And because the animal makes mistakes, there is some degree of dissociation between what you present here and the choice. And so the two boundaries that in respect to neural activity that decode sensory information of decisions may be different. And essentially the alignment essentially between those two boundaries from this alignment, so how similar they are, you can compute how much the encoding of sensory information is used to produce choices. If the two boundaries are perfectly aligned, then all the encoded information is used to produce choices. If, like in this case, they are misaligned, then some of this information is not used. For example, those trials here could be, those are blue stimulus trials, but are not well read out. So essentially, in a simple way, it uses from this trivariate model of stimulus, decision, choice, and neural activity, it extracts the boundaries in each space and then compares them, and you see how much of the information essentially encoded with the sensory boundary can be extracted with the decision boundary, which is not necessarily the same. And in the talk, I will use both. I think in the data that we use, those concepts, I mean, those algorithms are different but related, but they give the same results. And I will try to illustrate all of this with one application to one question about neural activity, which is a question that has tormented people for decades. So, like I said, since people started recording simultaneously the activity of several neurons, which started to happen at the end of the 1980s and beginning of 1990s. So people first started recording one neuron at a time and then started recording two, three, four, 10, now thousands of neurons. A common discovery is always find that, especially if you record neurons reasonably in the same neighborhood, in the same network, the activity of one neuron does not only depend on the external variables, but depends also on the activity of other neurons. So the neurons are functionally correlated. Correlation of the activity of neuron 1 fluctuates with the activity of neuron 2. And those, I will see later, those are measured, those are, essentially are taken as measure of the interaction between the neurons. And one question that people ask is, do these correlation help or hinder the accuracy of perceptual decisions? So is it good to have those correlations, or are those because they encode a lot of information, or are those a limiting factor? And I try to use these to address this long-standing question, which, like I said, has tormented people for many years. And just to gain to see what those correlations are, say that you have here a recording of neurons at the same time, so simultaneous recording of different neurons, and from this time series you can get the spikes. And essentially, correlation means that the activity of neuron 1 in one trial depends on the activity of neuron 2 in the same trial, even regressing out the covariation due to common tuning to the stimulus variables, or genuine correlations. And how I will try to represent this as done in the literature, we just consider for simplicity two neurons, but you can think of this as a multi-neuron dimensional, high-dimensional space. And each dot is a joint response, for example, the number of spikes of neuron 1 and the number of spikes of neuron 2. And you color dots with different color depending on whether they were collected, these trials collected in response to stimulus 1 or to stimulus 2. For example, here this blue will be in response to stimulus 1, and this will be in response to stimulus 2. And if the two distributions are not overlapping, it means that there is perfect decoding of the stimuli. If the distribution are overlapping, that when you decode the stimuli, you're going to make mistakes, or there is less information. And correlations mean like cigars, bananas, elongated distribution. Because essentially a correlation means that I can predict the activity of neuron 1 from the activity of neuron 2. For example, if I know the activity of neuron 1 is high during the stimulus blue, then I can also tell the activity of neuron 2 is likely to be high. If you don't have any correlations, the same diagram would look like this. Would look like essentially not elongated distribution, but circles within the same stimulus. When there are no correlations, I cannot predict meaningfully the activity of neuron 2 if I know the activity of neuron 1. And also, from what you can see from this diagram, and that in this particular example, having correlations would decrease the amount of information because it would increase the overlap between the two distributions. I will get back to this point later. And but also those are functional correlations, so computed just from the activity. We don't know the connection of the neuron, we don't know what they are. But also, I think that those functional correlations, at least within the local circuit, reflect to some extent, they give you some information about the local connectivity. To investigate this, we did a study together with Whaley and Chris Harvey at Harvard Medical School. And what they did, my experimental colleague did, they took a mouse that did one of these perceptual decision tasks and imaged the neurons with the calcium imaging, and then took out the brain at the end of the experiments, and they did electron microscopy anatomy. So for those neurons, about 200 and something neurons, we have the functional responses during the task, for example, whether they prefer left or right and so on, and a lot of information about their anatomy. And we can compute the functional correlation, but also we can compute post-mortem their functional connectivity. So we can compute whether the neurons are connected and the size of the synapses. So what we did was to take all the neurons that were connected and see whether the the functional correlation, so what is called here noise correlation, would be the functional correlation of the fixed stimulus, predict the size of the synapses between them. And there is a positive correlation of this quantity for the excitatory synapses and the negative correlation for the inhibitory synapses. So those functional correlations that we measure, you can interpret them to some extent as reflecting, at least to some degree, the actual essentially synapses between those neurons. And as an example of our application of our methods, we consider one task that was done by Chris Harvey that we analyzed in several papers. And it's a simple task, so mouse, and we use for this example here a posterior parietal area, but we also repeated the experiment in other areas. It's a sound localization task during spatial navigation in virtual reality. So the mouse starts and then goes through this linear corridor, and while the mouse runs, it gets a stimulus from the left or from the right. And when it comes at the end of this, it has to turn toward the direction of the sound to collect a reward. and we record the population of neurons, about 50 neurons per session. And if you look essentially at the trial average activity here of some of these neurons pulled across several sections, what you see is that those neurons essentially respond in a way that is selective to the choice or to the stimulus. These are correlated, but they respond transiently, so they respond only at a certain position during the maze. And so essentially, to type the task, you really need the population code because there is no neuron that essentially covers the entire task. And what is important for us is that those neurons, it's true that they encode at the individual neuron level sound location by transient activity, but also with long lag correlations across time. So within the same trial, if these neurons fire a bit more, also the next neuron will fire a bit more. If this neuron fires a bit less, those neuron will fire a bit less. This is what those correlations say, and this lasts several seconds. So you can think of this not as a sequence of uncorrelated neuron that fire or not, but really like a chain that propagates, so correlated activity. And one question is, what's the role of these functional correlations in forming accurate perceptual decisions? And like I said, the traditional way to look at this would be to look at this from the point of view of encoding information about the auditory stimulus that you need to discriminate. And if you want to understand what correlation between the neurons do to this, what you can do is to construct a decoder that decodes whether the stimulus, auditory stimulus was presented from the left or from the right, decoded from these, I don't know, 50 neurons. And you do it with the real data that contains the single cell response, but also the real within trial correlation between the neurons. And then you can do the same, but with shuffled responses, so shuffled fixed stimulus that have the same single neuron properties, but where these within trial correlations are gone or destroyed analytically. And if you do this, so if you destroy correlation, the kind of disappointing results that is pretty common in the literature is that you get more information. So it's like if those correlation would limit, would destroy information. So it would be better not to have them. You see them all the time, but then you say, what do? They just decrease your information. They increase the redundancy. And the reason mathematically those have been studied by various papers, like Diane and Abbott also did work at the same time. So essentially, the idea is that if you have neurons with, so if the correlations have, oops, sorry, if the correlation between the neurons have the at fixed stimulus have the same sign as the difference between the mean response to the stimulus. For example, here you have two neurons with both low response to the stimulus one and high response to the stimulus two, which will be what we call positive signal correlation, so similarly tuned neurons. And then if you have positive correlations aligned this way, then having correlation would increase the overlap between the two. And the same for the opposite, so negative correlation with anti-tuned neurons. And essentially, if you look at the structure of this data, you see that those two configurations are much more common than those. And because of this, those correlations are bad. So the kind of disappointing result, and is not by this study, but this has been replicated many times, is that those correlations from the encoding point of view they tend to be bad. So they reduce the amount of information that you can encode because they make the code more redundant. So you will see, it would be better not to have them. And then you ask, why do we have them? And we were puzzled by this, and one thing that we noted was that if we
Sorry,
computed, yes,
can I ask questions the previous thing? So,
please.
so you find that the creation is very important, but I think the absolute value of the so fraction correct. It's not so
Yes.
super so strong so difference.
Yes, yes. So 50% would be chance level. So they contribute 10% above chance, and with 50 neurons with recorded with calcium imaging, so they don't necessarily have high activity. This is pretty standard stuff. So a neuron essentially on average gives about 0.02, 0.03 bits of information. So when you put 50 neurons together, you get about 10% above chance. In order to get 2% correct, similar to the one of the animal, you would need some thousands of neurons. which
Mhm.
also makes sense because, this is a subsample of the neuron that participate to the choice.
Okay, I understand. And so let me check.
Yeah,
You are so observing only the PPC. So this is the PPC
yeah,
brain region, right?
yes. And
Okay.
one reason why we focus on PPC, sorry, I didn't say, is that it is described as an association area, for example, in the classic paper by Richard Anderson. So it's an area that is supposed to be at the interface between essentially sensory and motor, so decision function. So it's an area where looking at essentially decision making, also at this encoding and readout of information makes sense. But
Mhm.
we also repeated the experiments in other dorsal area like retropineal cortex and also in sensory areas. And I will tell at the end what's the difference between these association areas and sensory areas. Okay.
Okay, thank you.
Okay. And so what we noted is that if you compute correlations when the mouse makes correct decisions and when the mouse makes bad decisions, the correlations are stronger. We repeated this across many experiments. When the mouse makes correct decisions. And if you thought that those correlations would hinder the performance because they have less information, we would expect the opposite. You would expect that when you have incorrect behavior, you have also higher correlation levels. So those correlations seems to be helpful if you just look at this analysis. So how come? Why is this? And we made the hypothesis that maybe we can reconcile those views that correlation maybe they decrease the information, but they increase the performance or the useful performance if correlations help the readout of information. So if essentially maybe you have less information, but if you can transmit it better downstream, then maybe it's useful to have some levels of information. And there are many papers from the time of Christoph Koch and also Emilio Salinas and so on that made the point that correlation may help downstream propagation essentially because the neurons have a short integration time constant, so correlation pack the spikes in time. And also there are dendritic nonlinearity, so there may be some advantage of packing also spikes in space, which correlations do. And so in the end, correlation may increase, uh, so for any bit of information that you encode, you may transmit more of it downstream. And that's our assumption, but we wanted to try this assumption on the data. And how can we do it? And the way we thought we could do it is to compute what we call the readout map from the actual data. Readout map for us is the map from the information about the stimuli encoded neural activity and behavior. And essentially, I'll try to explain with a cartoon what is our hypothesis and then see how we computed it from the data. So first hypothesis will be that the readout is like the Bayesian readout, that essentially the more information you have, the more information you can extract. So if the readout is like the Bayesian readout or Bayesian readout plus noise, then what you would get is the following. I have to explain. So here I make again this cartoon with just two neurons and the ellipses are responsive to one stimulus and the other stimulus. And the case with the ellipses, the bananas, is the correlated case, the real case, and this is the same with the same single neuron property, then correlated, but when correlations were set to zero by shuffling or whatever the round. And what you can see here is that you have more overlap between distribution and correlations than here. So correlation would decrease the information about the sensory stimuli. So if you do the shuffling, correlated versus shuffle, you would have, like in our data, more information when you shuffle than correlated. So here correlation decrease the information encoding. If you assume that we have a map that is like a Bayesian decoder, it means that essentially what matters here is the total information. So the entire space of information is decoded the same way. It's just enough, the more information I have, the more I can use. So this readout map is uniform across the entire space. If you do that, essentially, the more information you have a Bayesian decoder, Bayesian plus noise, the more information you have encoded, the better will be what you can extract. And so the prediction is that you would have less fraction correct, so less behavioral performance if data is correlated than when data are shuffled. And also you would have weaker information, weaker noise correlation in correct trials than in error trials, which is the opposite of what happens with our data. So this kind of situation could be rejected based on our observation. And then we made the assumption because maybe those guys, those correlations help the downstream readout. This would work better, the readout, if the two population provide redundant information because correlation increase the redundancy. And essentially, a readout like this so would be something where when the two neurons give consistent information, redundant information, the readout is better. And when the two neurons give inconsistent information, the readout is less good. So for us, the quadrants in the space of neural activity in which um, the neurons give consistent information when they both decode the same stimulus. If I'm in this quadrant, low activity for both neurons would indicate that this is the blue stimulus. If you are here, both neurons would indicate that this is the yellow stimulus. If you are here or here, one neuron would indicate the yellow stimulus and the other neuron would indicate the blue stimulus. And our assumption is that if the readout is increased by correlations, then those guys here where there is more redundancy, these responses are better read out. So any bit of information here is better read out than the same bit of information here. So that's our assumption. If you implement a readout like this in a simple model, then you would expect to have better behavioral performance with correlations. So correlations are useful. And also, like we find in our data, more strong correlation in correct trials than in error trials. And the reason why here you would have with this readout, which is not optimal, but is essentially a suboptimal readout that we think would happen in the brain, why you have better performance here even with less input information is that correlation they increase the number of trials that are difficult to decode, which is the overlap between the distribution, but they also increase, if you compare this with this, the fraction of trials that are better read out. And so what we wanted to do is this is a simple conceptualization, but can we test this on the data? So what we did, we take essentially all spike trains, all decision from all mice and so on. And in each case, we construct a Bayesian decoder of the stimulus, which essentially construct the encoding. So this is essentially for us the best reconstruction of the posterior probability of the stimuli given neural responses. And then we say, can we compute how these fluctuate with the actual choices of the animal? So we made essentially logistic regression of, I spare you some of the detail, but essentially there was one term that would be just essentially proportional to the stimulus which is decoded optimally from neural population, so essentially the posterior. And if the readout by the brain was Bayesian plus noise, then this would be the early term. But then we did an interaction term that essentially makes it easier or more difficult to read information depending on whether it's consistent between the different neurons or it's inconsistent. And we left essentially these terms here, just all are like essentially they fit to the data. And what we found is that you have those terms, so you can get better predict the choice of the animal if you have those terms. So this is not just Bayesian plus noise, but there is an interaction essentially between how individual neurons encode information, redundant or not. And the signs of these coefficients are exactly like in our little conceptual cartoon. So the sign of these coefficients say that this readout map you have a better probability of decode this given bit of information if both neurons or both pools of neurons, if you do at the population level, give the same consistent information than when you have the same bit of neurons, but they give inconsistent information. And this is a suboptimal operation. Also, this also implies that at least measured in this way, the readout of the brain is suboptimal. Optimal would only use the total information. Suboptimal, instead, if you use the consistency between the different sources of information, is suboptimal. An example is if you want to learn about computer science and you have two sources, one is Jeff Hinton and another is me. If you are an optimal readout, you would only listen to Jeff Hinton and would ignore me. But if you know nothing about computer science, you are suboptimal. then if by chance I confirm what Hinton says, maybe you will take it more seriously. That would be a suboptimal operation, and this is what those neurons seem to do. So essentially, if there is some consensus within the population, any bit of information has a higher chance to make it downstream to inform the choice. And
Let me check through basics this equation. So S is a sort of sensory input, and S hat is also decoding sort of sensory input from the neural
yes,
activity. Yes, and I don't understand the S hat so times the correlation, then one minus S hat. The Y sort of one minus S hat sort of term is necessary.
yeah, this is just to compensate because it is a binary stimulus, and so
Binary
if you were
stimulus.
to, yeah,
Okay,
the
okay.
S is a binary stimulus, so it's left or right. So for the binary stimulus, essentially this becomes this, that if those signs are positive, then it increases readout in this region and it decreases readout in this region. I mean, it's just a little trick to express it in a simple way.
And
And it can be also asymmetric within the stimuli. But in practice, I think if you look at this, there is a bit of asymmetry. And the reason I think there is a bit more noise from the microscope from one side than to the other side. So, but okay, it's a small technical.
And so the correlation is a sort of previously COR. So COR is a correlation, right?
So is a consistency. So it's computed
Consistent.
as consistency. So essentially, it's when they decode the same stimulus. The reason that correlation is defined across trials. So
Mm.
correlation is the property of all trial, but you want a single trial measure of correlation. And for us, the single trial measure is because correlation increase the redundancy, so increase the number of trials in which the both neurons give the same information, like this.
Uh-huh.
Then essentially what we do, we measure in each trial the consistency that is are the neurons in the population encoding the same stimulus. This is consistent, yes or not. So we turn it into a binary variable. So essentially it's a way to say this readout is based on this single trial property, which is consistency, which is modulated by correlations. And that's the way essentially we do the calculation.
Mm.
I
Okay.
don't know if this is clear. Okay. It's a way essentially to express single trial correlation. Okay. And we don't have a way yet to really essentially modify the correlation on the fly from the neurons and then see what is the output. But what we can do analytically is that we can test whether this beneficial effect of correlations or consistency for readout would mean that the brain works better with correlation than without. We take essentially the real data, which are correlated, and then we feed it to the decoder. Sorry, from this readout model on your activity, we can generate choices with the best algorithm that describe the choices of the animal based on neural activity, and we compute the predicted task performance due to recorded correlations. And if you do that, we say that this total population contributes about 3.5% correct above chance to the total essentially performance of the animal. The animal does 75% correct, so you would need more than 50 neurons, but the neuron contribute this. But then if you shuffle the neuron, you have more input information, but less of it is used, and so essentially you would have lesser task performance if and also stronger correlation in error trials if there were no correlation. So this suggests that you have with correlation a disadvantage in encoding and advantage in readout, and if you put them together, the advantage for readout overcome the disadvantage for encoding, which also means that there is an advantage of having some non-zero moderate level of correlations in the brain. And if we did the same experiments, but in sensory cortex, with a similar mechanism, but with overall weaker correlations, and so less encoding damage, but also less advantage in readout. And our interpretation of this, I don't know if it is correct, but is our interpretation is that if you are in a decision area, then it's better to have a stronger correlation because you don't need to encode so much information, so you can afford some reduction because you encode only the task relevant information. And so you don't need really to worry too much about the problems for encoding, but you really need a good readout because when you take a decision, you want to make sure that it is implemented downstream. Whereas in sensory areas, there is a higher premium of encoding more information but maybe you can lose, you can afford that some of it is not read out. And so that's why maybe we observe consistently across experiments stronger correlation in decision areas or association areas than in sensory areas. That's our theory. I mean, just a speculation, but that's what we found. And this is just based on observation, but I think a good thing about this theory that it gives a prediction for experiments. And my colleague Chris Harvey with Dan Wilson is developing a technique when he tries to essentially both perturb with single cell resolution and image the neurons. And if you do that, at some point you can also essentially change the correlated patterns and so on. And our prediction is that if you inject into the brain by stimulating cells the same bit of information, either in a consistent or inconsistent way, you would have a better readout of what you inject into the brain if it is consistent. For example, if you stimulate a neuron that gives a small amount of information in a consistent way, it would be better if you stimulate a neuron that have a lot of information in one way and the neuron that has the opposite information. This is something that we are trying to do. We are not there yet, but we did a vaguely similar experiment in a bit more simple way with the Marienberg in New York. And essentially, there was an experiment without any sensory stimulus. So the mice were trained to recognize a given stimulation pattern from a random pattern. And by varying, it's a long story, but essentially independently the dimension of the random pattern, you can construct some of these readout models similar to those, and they have interaction terms that tell that essentially the correlation between the different times at which you activate the different populations, if you do it in a correlated way, there is a better or a different probability that the mouse can recognize the information injected into the brain that would support this idea that essentially there are interaction terms, so the readout is modulated by correlation. Okay, that's one. So this is something that can lend itself to optogenetic also tests, but also can be applied by essentially looking at one area and then try to infer how much of this area has been read out or this information in the area by looking at the downstream behavior. But then you can also try to ask a finer question if you have instead data that have additional ingredients. And one thing that we started to think about is that if you look at a lot of studies about neural coding, I think almost all of them are based on population of neuron defined as neurons in the same neighborhood, say neurons in visual cortex, neuron in posterior parietal cortex, in this field of view and so on. But I mean, this is in some ways a good way to define a population, but another possibly more biologically plausible way to define a neural population is within the neuron that I record, I consider as population, as group, neuron that project to the same downstream target. For example, here I might say that a population is the blue neuron that project to this downstream target. Another population is the neuron that project to this other downstream target. Another population is the red neuron that project to this downstream target. So we thought that one better, maybe an interesting way to look at population code is to define population code that those group of neurons within a neighborhood that project to the same target and see whether the neurons that project to the same targets are organized in a different way in terms of their interaction with respect to neurons in the same neighborhood, but they don't project to the same target. And so essentially what Chris Harvey did, he designed an experiment like that where he put retrograde tracers with some color, virus, and essentially they travel back from the target area. This is posterior parietal cortex, and it would be a target area. I think one was retrospinal cortex, one was contralateral posterior parietal cortex, and another was anterior cingulate. So those are three targets, downstream targets of PPC. And those virus travel back and they give a little bit of color. So the green color is the one for the activity of the neuron, the G-camp. And the blue hue that you see here is the color given by the virus that travels back. For example, say this neuron here and this neuron here would be neurons that don't project to any of those targets because they were not marked by the color. But this neuron with the blue halo here and this neuron with the blue halo here, and this one, those guys would be neurons that form a network that project to the same downstream target. And so what we did, we started to looking at with these techniques, sorry, I will have another 5 minutes. Is that okay to conclude the talk? Or yeah, I hope because I'm a bit late. It's almost 1 hour. Can I go on for another 5 minutes or? Yes, okay. So one thing that we noted by considering either neuron that project to the same target or neuron that don't project to the same target, so they don't get any label, is that correlations are stronger, and especially in correct trials for neurons that project to the same target, even if they are not close to each other. And this, I think, confirms or is compatible with our view that those correlations are really key for downstream information transmission because they are stronger in the neuron that project to the same target downstream and especially during correct behavior. So one, that correlations are important for downstream transmission, and second, that this is important for the accuracy of the task. And then also we started to look at another thing that we think is important is to try to look at the difference in the structure of this network that either project or don't project to the same target. And so to study this, we essentially characterize each pair of neurons as either having correlation that increase the information or decrease the information. And I think you would have more minus than plus at the population level in many experiments. Sometimes you have the opposite, but one thing is not only how much you have, but how they are structured. For example, you could have that you have synergistic triplet essentially of all neurons that are connected to each other, that form hubs and then or pools, and then those are more connected within the pool than outside the pool, or it could be also the other way around, like more redundant networks that are have more essentially redundant connection between each other, the pools with synergistic connection between them, or a random network and so on. And what we did was essentially to compare the distribution of these triplets, which is an information of the network structure, higher level network structure, in the real data compared to the random network that you obtain by essentially placing the same plus and minuses links here, but just in a random way. And if you look at the neuron that don't project to the same target, they have a completely random structure. But if you look at the neurons with um, the project to the same downstream target, you see that the triplets whose product is plus, so minus, minus, plus, 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 are more numerous than the triplets whose product is minus, 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 plus, plus, minus. And we thought, why is that? And to start to understand that, we did some mathematics where we took the total information computed by the population and making some small assumptions justified by the data, so that the single neuron information is relatively small, like I said, and also that the level of correlation is relatively small, which is also true. Is present non-zero but small, then you can do like essentially some kind of Feynman diagram, Taylor expansion of this big information, is like essentially doing Feynman diagrams in physics, and you find that those essentially the various triplets have in this term structure. So the structure of the network contribute to the information in a way where the triplets whose product is plus contributes positive information, and the triplets whose product is minus contribute negative information. So essentially what you have here in this structure is that the triplets, the neurons are organized like this way. Essentially would be this, so synergistic neurons connected in the pools and with redundant interaction between the pools, and essentially such that you have more information, sorry, more triplets of neurons that increase information with respect to triplets of neurons that would decrease information. And so essentially you have a network structure, microscopic level, which is present only in the neurons that project downstream and is present only during correct trial. So our interpretation is that there is a structure of information encoding at the local level, which is done to promote downstream transmission and is crucial to do the task because it's absent in error trials. So that essentially is the way. If you look at those neurons, if you label the neurons, you can get more information about the meaning of the network organization and the network encoding that you don't see if you don't isolate the neuron that project together to the same target. And the final thing in the last couple of meetings, sorry, I started talking, so I went a bit over, is that we also analyzed this problem of in some experiments you may have simultaneous recording from different regions. For example, you have your recorded region, but you can have a downstream region. And suppose that they are hierarchically organized, then it would make sense to consider that you have some information encoded here. Could be sensory, could be motor, whatever you like. And then this information from this activity transmitted here. And we ask the problem is, can we characterize the transmission of information from here to here, but only transmission of information about the stimulus? And we look at available methods. You can do this essentially by looking at the time series. Something that works very well is called transfer entropy, which is the nonlinear version of Granger causality, where you take the two time series, and what you do, you compute essentially the total information transfer from X to Y, which essentially is computed as the information that you have in the future of Y, so the putative receiver, um, about the past activity of X, but that was also not present essentially in the past of Y, so it's new information. And that's essentially the Wiener-Gansen principle. But this is total information, doesn't tell how much of this transfer is really transfer about the variable you're interested in. So we try to do something like this again with this method of information theory. To cut long story short, we define this feature information transfer from X to Y as the total amount of the information about the stimulus that you have in the future of the putative receiver that was present exactly in the same format in the past of X, but was never present, so unique to in the past of Y. So it's something that I think makes a lot of sense. It's a bit complicated to compute, but my former student Marco, who is now a postdoc at MIT, did it. And we have this measure that essentially, rather than computing the total information from X to Y, tells you the information from X to Y that is transmitted specifically about the variable of interest. And sorry if I overrun, we started also applying the two experiment, and I have time only to show just some highlights. So with a colleague coming from UCSF, he did an experiment where he recorded from two brain regions, the motor cortex and dorsolateral striatum, as a rat was learning a very fine grasping skill. So essentially the rat has to learn to collect and take a very, very small pellet of food and to eat it. And at the beginning, the mouse makes a lot of mistakes and the grasping is not so good. But then as the mouse learns, it becomes very good at doing this. So it's essentially recording of those two regions and the fine grasping skill. And to cut a very long story short, if you look at this problem of essentially how those two areas, the motor cortex and the striatum, communicate, if you look just at the oral information propagation, the coherence and so on, if you compare the case at the beginning where the mouse is not skilled, those areas communicate, interact bidirectionally, but not very strongly. And if you instead consider, do the same measure of, say, transfer entry, total information transmission, overall information transmission, at the end, both of them interact very strongly. But the question is there a change in information direction? If you just look at the total information, how much they talk each other, you don't see any difference. But if you look instead of the information about the kinematic variables that are important to do the task, then you have at the beginning, essentially, there is more information flow from motor cortex to the striatum, and then at the end, there is more information flow about this fine skin variable from the striatums to the motor cortex. So the primary direction is the reverse. So I'm not a motor cortex or motor learning specialist, but to me this is interesting because it shows that there is some merit in looking at those methods because the overall transmission of information between brain areas is somehow dissociated from the specific information transmission about different task variables as you learn to do the task or if you do the task and so on. So I think this suggests there is some worth in the method that we presented. And if I have to recapitulate, essentially our work as humble theoreticians, but trying to contribute to the progress by science in providing methods is we did quite a lot of work over the last few years to provide analytical tools that dissociate the contribution of encoding and readout of information neural activity to form perceptual decisions. And we try to design them to adapt them to several experimental conditions, which include recording from one area with behavioral choices, recording from one area, and also having information about the neuron process with the same target, recording to multiple areas. And applying developing this method, we've always found, I think, reasonably interesting results. And to me, what matters most is that there will always, in all the data we analyze, some kind of dissociation between encoding and readout. So this suggests that it's not enough to study only encoding in a given area, or to study separately encoding and readout and try without thinking about how they would combine. And also those studies that we have suggest that neural codes may be organized not to optimize information locally, but rather to optimize the information that is made available for downstream use. And we have examples like in parietal cortex where this correlation essentially don't promote local encoding, but they promote downstream transmission of information, and so on and so on. And also something that I just touched briefly upon is that one good thing is that this framework can be also used to make very specific hypotheses when you are able to manipulate individual neurons. And so essentially can be used as we're trying to do now to design perturbation to test hypotheses in a causal way. And I know that this is a series of seminar about digital brain models. So I hope that this seminar will be somehow interesting also to the people that work on them. And what I believe those methods could be interesting for is that if you have a digital brain model and you have some essentially very complex dynamics because you have many neurons areas and so on, and then you want to understand essentially characterize how the various architecture that you put in change information transmission, or what parts of the digital brain model are more important to generate your simulated behavioral output and so on. The tools that we did could be also useful to somehow gain some understanding about this. And this is it. And I would like first to thank you for your attention and for bearing with me for this 1 hour. But also again, I thank all my postdocs and students that did all the work, and also all my collaborators. And thank you very much for your attention. I appreciate it.